Hi micro majors, my name is Mateo and I'll be guiding you through the experiments in the mycology module. So to start off, we'll be collecting fungi first. I'll be showing you how to collect fungi with the provided PDA plates. PDA stands for potato dextrose agar, which is an all-purpose growth medium for fungi. You'll take these plates and place them in an area where you want in your home to see if there's fungi or not. Once you've chosen a spot, you want to open the plate up and let it be exposed for about 3 to 6 hours. After those 3 to 6 hours, you want to close it. And then with your other plate, you want to repeat the process, but you might want to choose a different place. But once you've collected your fungi, they should grow, should look something like this. And you're going to want to seal it. To seal it, you're going to take a parafilm strip. Once your plate's sealed, you want to make sure you let it incubate, but you want to make sure it's completely sealed. So to do that, you'll take some parafilm, and what you'll do is you'll put one of your index finger on it, and then you're going to wrap it around the plate just very slowly. And if one isn't enough, you can get another one and just put it over and complete the circle. Once your plate's sealed, you're going to invert it downward so that the media is on the bottom of the plate is upward, and then you'll let it incubate for however many hours. And after those hours, you'll have some fungal growth. And make sure once the plate is sealed that you don't open it back up, but just take note of the various different colors and sizes and morphologies of the fungi. All right, so for the next part, we're gonna get into how to stain fungal samples that you want to view under the microscope. We'll use a stain called lactophenol cotton blue, which stains the chitin, which is the material that makes up the fungal cell walls. To begin, you're going to want to take a piece of fungi from your environment. It can be, for instance, this piece of moldy ginger, or if you're a little bit extra, I don't know, you can take it from insects that have mold out there, which are interesting. But for simplicity, we're just going to use this piece of moldy ginger. All right, you're going to get your transfer pipette. And you're going to take 1 drop of cotton phenol blue, and you're going to place it on the side. All right, once your drop is on the side, you're going to take a piece of Scotch tape, which is provided. You're going to rip it out. And then what you're going to do is you're going to fold it over just a little bit, and you're going to choose an area on your fruit or whatever you decided to take, and you're going to go for a desired colony. So for here, we're going to go for this colony. As you can see, it picked up some fungi. And then what you're going to do is take the piece of tape, make sure the side that has fungi is down, and you're going to put it on the side where the drop is. Once that's done, you're going to take a cover slip and also put it on top. It's possible that it might not be needed, but and now you're ready to start viewing your fungal samples. Just make sure to take note of the different microscopic structures, the different colors. If you're trying to identify them, you really want to look at the spores and the fruiting bodies. But yeah, fungi are very interesting, and I hope that you learned something from this introduction to fungi today. So good luck, Gators.